Hello, ladies and gentlemen. Hello, everyone. This program topic is proteasome inhibitors, and I would like to use this condition for nuclear factor kappa B, transcription factors that nearly 200 genes expressed some pro-inflammatory factors, cytokines, prostaglandins, nitric oxides, that these molecules responsible among macrophages and cancer cells strong inflammatory dynamics, carcinogenesis, cancerogenic transformation, energy suppression, inhibition, normal DNA and its dynamics, silence, and genes silence, and activating foreign dynamics, non-self dynamics of cancer cell, mutations grow identity. So very important and vital role actor, medicine, drug, proteasome inhibitors. Capping a decades-long research effort in the ubiquitin proteasome field, the 2000 some activity was awarded to some scientists work on elucidating the cellular mechanism of protein degradation. The ubiquitin proteasome pathway is responsible for the degradation of diverse cellular protein with exquisite specificity and has emerged as a central player in the regulation of diverse cellular processes, given its key part of maintaining cellular homeostasis. Protein degradation may seem an unlikely target for pharmaceutical intervention. However, the discovery that several anti-tumor natural products exert their action via proteasome inhibition provided a rationale to develop proteasome inhibitor drugs for cancer treatment. As a result, proteasome inhibitors from both natural and synthetic origins have been intensely pursued as therapeutic agents, and to date one proteasome inhibitor, bortezomib, was approved in 2003 for treatment of multiple myeloma and more recently for mantle cell lymphoma. Immunoproteasome, an alternate form of constitutive proteasome, in striking contrast to constitutive proteasome, the immunoproteasome is restricted in its tissue distribution to cells of hematopoietic origin. In certain pathological inflammatory conditions, the immunoproteasome is upregulated in non-hematopoietic cells by the action of inflammatory cytokines, including tumor necrosis factor. Despite recent advances implicating the role of immunoproteasome in certain pathological disorders such as neurodegenerative disease and diabetes, the physiological role of immunoproteasome is largely unknown, with the exception of the function in major histocompatibility complex class 1 antigen presentation. The major problem that limit further understanding of immunoproteasome biology is the lack of appropriate molecular probes that selectively target the immunoproteasome catalytic subunit. Proteasome inhibitors, natural product proteasome inhibitors, models of action studied have revealed that several natural products are selective and potent proteasome inhibitors. For example, lactacystine 1 is a Streptomyces lactacystinaeus metabolite that was discovered on the basis of its ability to induce neurite outgrowth in the murine neuroblastoma cell line neuro2A and was subsequently shown to target the 20S proteasome by an irreversible modification of the amino terminal threonino beta subunit. Moreover, in an effort to search for anti-tumor agents displaying specific activity against B16 murine melanoma, alpha peptide alpha beta epoxyketone epoxomycin from unidentified actinomycet strain. Epoxomycin specifically targets the 20S proteasome, but unlike other classes of proteasome inhibitors that show non-target specificity, epoxomycin is highly specific for the 20S proteasome as revealed by the crystal structure of the yeast 20S proteasome complex with epoxomycin. This unique specificity of epoxomycin is a result of six-membered morpholino ring formation between the amino terminal catalytic THR1 of the 20S proteasome and alpha beta epoxyketone pharmacophore of epoxomycin. In addition, eponemycin and anti-angiogenic peptide alpha beta epoxyketone isolated from Streptomyces hygroscopicus and continue and detailed. That's enough for proteasome inhibitors in cancer. Clinical activity of bortezomib. For administered bortezomib rapidly cleared from circulation intravenous administered bortezomib is rapidly cleared from circulation, although the pharmacodynamics effect of bortezomib inhibition of proteasome recovery with a half-life of approximately 24 hours. Ascending doses of bortezomib inhibit proteasome activity in blood in the dose-dependent fashion of reach a maximum of 65% inhibition at a maximum tolerated dose. Bortezomib is typically administered on a 3-week cycle on days 1, 4, 8, and 11. Daily dosing schedules have been associated with served toxicity in animal studies. Bortezomib has shown remarkable single-agent anti-tumor activity in wide range of hematologic malignancies, including multiple myeloma, non-Hodgkin lymphoma, Waldenström macroglobulinemia, and other diseases. Bortezomib was approved for treatment of multiple myeloma based on a single agent objective response rate ORR of 28% in a phase 2 study in 193 heavily pretreated patients who were refractory to the most recent therapy. Grade 3 reversible thrombocytopenia was observed in 28% of patients. The rapid reversibility between dosing cycle together with the observation that enlarged megakaryocytes were present in bone marrow suggests that bortezomib mediated thrombocytopenia results from inhibition of platelet budding. Neuropathic pain was observed in 12% of subjects requiring immediate dose reduction or discontinuation in order to avoid irreversible damage. Other toxicity included nausea, vomiting, diarrhea, constipation, neutropenia, and anemia. Bortezomib has been tested in variety of solid tumor in phase 1 and 2 studies using variety of dose and schedule of administration. Partial responses were reported in 8% of patients with refractory non-small cell lung cancer. Although the TTP was 1.5 months, these encouraging results in NSCLC have led to testing of bortezomib in combination with other chemotherapeutic and with partial responses, decreased prostate-specific antigen level, and lymphadenopathy in few patients. Have also been observed in patients with hormone-independent prostate cancer. Stable disease has been reported in patients with hepatocellular carcinoma, metastatic melanoma, neuroendocrine tumor, and advanced renal carcinoma. These drugs dosage schedules, routes, had been expressed and later will be again dosage. Clinical development of other proteasome inhibitors, carfilzomib, PR171-8, entered phase 1 clinical development in 2005 based on preclinical results showing tolerability of repeated daily doses of this compound. Carfilzomib is being investigated for the treatment of hematologic cancer using two different schedules of administration. In one study, carfilzomib is being given daily for 5 days on a two-week cycle. In the second study, patients received 2 daily doses of carfilzomib weekly for 3 weeks on a 4-week cycle. Partial responses have been observed in patients with multiple myeloma on both protocols. Reversible thrombocytopenia is seen with carfilzomib, similar to bortezomib. There have been no observation of neuropathic pain thus far in the clinical trials. The third proteasome inhibitor, the lactacystine analog MPI-0052, has entered phase 1 clinical development for the treatment of hematologic and solid tumors. This compound has a similar potency of bortezomib and carfilzomib against the chymotrypsin-like activity of the proteasome, but has been shown to have oral anti-tumor activity in preclinical studies. Initial clinical trials are utilizing an intravenous formulation of this compound. Proteasome inhibitors will be used for nuclear factor kappa B blocked inhibition, but never impossible that this drug, bortezomib, carfilzomib, and so on, never impossible that sole therapy, treatment and cancer disease alone should be main cornerstone. Nuclear factor kappa B drugs, proteasome inhibitors, histone deacetylase inhibitors, hypomethylating agents or DNA methylating inhibitors, cortisones, non-steroidal drug, non-steroidal anti-inflammatory drugs, high energy phosphate compound, some critical oils, glycosaminoglycans, and some doxaenoic acid, eicosanoic acid, and some very important role together group actor should be possible. Any stage completely totally cancer disease solved treated therapy. And stop this point at this point, and we will be continued other actors. Thank you very much. Thank you so much. See you soon.